All right, welcome everyone, and thank you for joining us today. My name is Angela. I'd like to welcome you to our webinar titled Improving Speed, Efficiency, and Resilience in the Development of Biopharmaceuticals with End-to-End Bioprocess Digital Twins. Before we get started, I just want to go over a few logistical details with you. So this is what the 23 webinar platform should look like on your end, with the screen on the left-hand side where you're currently seeing the presentation. And on the right-hand side here, you should see the questions panel at the top. This is where you can ask questions for the speaker, and then at the end of the webinar, we will have a Q&A session where we will respond to those. If you are joining us from a mobile phone, you should see a similar interface, but the questions box will be at the bottom and the screen will be available at the top for your viewing. And as I mentioned, please submit your questions for our presenter anytime during the webinar, so we'll have some time to go through them before the Q&A session at the end. Okay, and now I'd like to introduce our speaker for today. Dr. Edward Close is the head of the Bioprocess Modeling Competency Center at Siemens Digital Industries Software. He has an engineering doctorate in biochemical engineering from University College London in collaboration with Pfizer. Edward has over 10 years' experience at Siemens, leading the development and application of Siemens mechanistic and hybrid modeling solutions to deliver value across the pharmaceutical industry. Okay, so with that introduction, I'm going to go ahead and hand it over to Ed to get started on today's presentation.
Okay. Well, thank you very much for the introduction, and thanks a lot everyone for joining. So welcome to today's webinar. So you've seen the title already. So let's get going. So let me start by asking a question. So why bioprocess digital twins? So what we're hearing repeatedly is the biopharmaceutical industry is rapidly evolving. There's growing patient demand, there's new modalities, and there's a continual pressure to deliver. So there's an ongoing need to bring high quality biologics to the market faster, more efficiently, with flexible and sustainable processes, whilst still meeting regulatory expectations and business objectives. And of course, this is a very complex and difficult challenge. And what we see is organizations are increasingly looking to digital transformation to help them meet these challenges. So what we've seen is there's been a huge increase in demand for digitalization technologies in recent years, and in particular, the use of modeling and simulation stands out. So this is reflected in communications from academia, from presentations you see at industry conferences, and also now from regulators. So this is a nice quote from the FDA, stating that in the last decade, modeling and simulation has become firmly established as a regulatory science priority. And this is a very nice publication from a few years ago, but if you go here, it kind of describes from their perspective successes and opportunities in modeling and simulation for the FDA. Industry agrees. This is a nice publication by industry leaders from 14 major biopharmaceutical manufacturers and suppliers. And it describes a remarkable agreement that they have on the problems to solve in industry and the significant opportunity to transform development and manufacturing through end-to-end integration and technology advancement. And this is really what we're going to be talking about in today's webinar. So at Siemens, we provide software, services, and online solutions that enable our customers to rapidly configure, calibrate, and deploy science-based, data-calibrated digital process twins. And I'm part of the bioprocess modeling team. I head up this team for a few years now. And before I go into all the details, I thought I'd first of all start with some basics. So you may ask, what is a bioprocess digital twin? So for us, bioprocess digital twins are comprehensive virtual representations of the bioprocesses that are utilized across development, engineering, and manufacturing to understand, to predict, and to optimize the physical counterpart's performance, to monitor it, and to facilitate decision making. And at Siemens, we have an end-to-end solution for bioprocess digital twins that is underpinned by science-based mechanistic and hybrid modeling approaches. So this is something that we've created in collaboration with big pharma partners for modeling the entire biomanufacturing process in a single platform that's used across the process lifecycle. So when we talk about end-to-end solution, first of all, we mean modeling the full bioprocess, so all of the different unit operations. And then secondly, we mean the ability to use those models integrated with each other to model the full bioprocess, and then to be able to take those models and use them across the process lifecycle. So why are people interested in this? So these bioprocess digital twins enable organizations to develop and produce medicines faster and cheaper by replacing physical experimentation with in silico modeling to design, to optimize, and to scale up biopharmaceutical manufacturing processes, and in the process reduce staff, material, and lab costs per project, which means that you have a more efficient organization and you can push more projects through the pipeline with the same resources. Then what we can do is we can deploy bioprocess digital twins into manufacturing for soft sensing, monitoring, and real-time optimization. And of course, this can then increase robustness, yield, and productivity. Oh, I think we have a little bit of noise on the background. Maybe that's you, Angela. Just before we jump into the first poll. So the term digital process twin is often used quite loosely. So I think it's useful to define a few categories that depend on the degree to which data flows from the physical object to the digital object. And so we have these three sort of categories: digital model, digital shadow, and digital twin. So with a digital model, the data flow is manual. With digital shadows, the data flows from the physical to the digital automatically, but the digital to the physical is still manual. And then we have the full digital twin where the data flow is completely automated. So what we tend to see in general is that in R&D and engineering, organizations are predominantly utilizing digital models and digital shadows. And then in manufacturing, it's more aligned with digital shadows and digital twins. However, there are certainly outliers. So for example, there are definitely cases where digital models are used by manufacturing to analyze deviations. And certainly I've had conversations where there's interest, at least, in utilizing digital twins for automated process development, where we have a back and forth between the experimental system and the digital twin, which is kind of guiding the actual experiments that are performed.
All right, so then we can go into our first poll. So we do have a few polls we're going to go show you guys throughout the webinar. So we'd appreciate if you guys would take the time to respond. So I just went ahead and started the poll. You guys should be seeing it now. And I'll leave it open for about 30 seconds or so for everybody to take the time to respond. All right, fantastic. So it looks like we got a lot of responses, so I'll go ahead and close that. And yeah, feel free to continue it.
Okay, so here at Siemens, we have customers that are utilizing digital process twins in bioprocessing, both offline and online. And so we have examples of digital models, digital shadows, and digital twins. So what we're going to do now is we're going to have a look at a number of examples. And the first thing we're going to look at is offline usage. And this tends to be where organizations kind of start their digital twin journey. Okay. So let's consider a bioprocess. So here we have a continuous monoclonal antibody manufacturing process. But what I'm going to describe in the next few slides is equally applicable to batch processes, and it also applies to the kind of range of modalities that we usually see across the industry, such as vaccines, other therapeutic proteins, gene therapies, mRNA, et cetera, So when we design these processes, unit operations are usually considered discretely, and this means they each receive their own set of control elements and acceptable ranges, which are then assembled together to provide an overall control strategy for the entire process. And I think historically, or at least over the last kind of decade, a lot of the applications where we see science-based mechanistic models, at least in biopharma, R&D, engineering, manufacturing, tends to focus on a single unit operation in the process chain at a time. And here, when we discuss this with our customers, we're always discussing sort of the advantages over a purely experimental approach. So being able to perform development tasks faster, cheaper, and with greater process understanding or fundamental understanding. So let's have a look at a few case studies here. So the first example is the design and optimization of a chromatography gradient solution. So this is a process that's producing a peptide and a oligonucleotide. And the step is one of the polishing steps that removes product-related impurities. And in particular, there's one which is particularly tricky. It's product impurity N-1. And this is because it elutes at a very similar time to the target. So we can also see here there's an N+1, a low charge, and high charge, which are also important. Now, optimizing this process experimentally required over 3 months and a significantly large experimental effort using a kind of traditional DOE approach. Now, in this project, a mechanistic chromatography model was calibrated using data from 3 gradient elution experiments. So these were linear gradients, and samples were taken every 1 column volume during the elution. And the concentration of each of the main species that we wanted to model was determined using HPLC. So this data was imported into the software and used to calibrate the adsorption isotherm part of the model. So in this particular case, we're using a rate model of chromatography. And the calibrated model was then used to explore the design space. And what we found was that the model was able to identify the optimal gradient in about a week. This was confirmed experimentally. Fascinatingly, the optimized gradient that the model produced was very similar to the experimental approach. But of course, it was able to identify that using significantly less material with only 3 experiments and in a much quicker time. So by introducing mechanistic modeling into development workflows, we're able to significantly reduce the number of experiments that we need to conduct, the time and the cost. Okay, so the second example is the optimization of the feeding strategy of a production bioreactor that produces monoclonal antibody. So this is a highly complex process, of course. There's lots of decision variables. In this particular case, there were 3 nutrient feeds that were fed daily and a temperature shift. So in this work, we start by configuring a mechanistic bioreactor model. And this includes a segregated cell population model where we're tracking multiple populations of cells. So we have viable cycling, viable resting, dead cells, and lyzed cells. And then we also have phenomena models to model the cell growth, the cell death, the cell lysis, the consumption of nutrients, the secretion of metabolites, and of course the production of the product of interest. So in this case, there were 22 experiments. So 7 experiments were used to calibrate the mechanistic model, and the remaining 15 were used for external model validation. And you're seeing here one of those, or part of one of those models that validation runs. So when you're calibrating a bioreactor model, we not only need to know information about the cells, but we also need to know the data on things like the key nutrients and any toxic metabolites. And so in this case, we're using 1 sample per day. So for the majority of the experiments, we saw very good predictions. But in this case, we identified in the sort of analysis of the model validation that the model was struggling with high seeding densities and with the impact of temperature on cell culture kinetics. And in this case, the data which we collected for the model calibration was something that had been previously generated for sort of a DOE. And so this wasn't data that was designed specifically for model calibration. So it's important to be aware of this. So for this phenomenon, these phenomena, we explored a number of options. So the first thing that we explore is can we extend the mechanistic model, introduce some additional equations that would be able to describe that phenomenon. But in this case, we decided that actually this was part of the process that we didn't have enough fundamental understanding on, and so we moved to a hybrid modeling approach. So hybrid modeling is something which is reasonably new, and it's one of the big reasons why you now see bioprocess modeling in industry, whereas previously we didn't see it as often. And that's because we can use a hybrid approach where we embed something like an artificial neural network or even a PLS model sometimes into the mechanistic model to supplement the mechanistic model approach. So in this case, we have a parallel approach. So the inputs to the data-driven model and the kinetic model are the same. So for both of these, we're sending the concentration of the nutrients and the metabolites, the cell concentrations to both the data-driven model and the kinetic model. The kinetic model, which is fully mechanistic, predicts the rates of things like secretion, of growth, of death. And then the neural network, which is coupled very closely to that model and produces a correction factor. And that correction factor typically is something which is quite close to 1. And we can see on this graph here that this neural network producing a correction factor. So using this approach, we can get a much better prediction for the difficult to predict experiments. And so here we can see good fitting is maintained for the runs that were used previously to train the purely mechanistic model. And then we've introduced some additional runs to be able to calibrate the hybrid model. So this is something where we have now got a much better fit, but of course this is a fitting, so we expect this anyway. On the right-hand side, you see though the external model validation or the blind testing, and you can see here that we found that the prediction capability has improved greatly for a high seed density run that could not be predicted previously. Then the validated model was used to explore the optimal feed strategy, and in particular in this case, we also considered the usual variability that we would expect to see in this bioreactor. So rather than a point prediction where we look at one input and then we predict the outcome, instead we use the expected variability in the inputs, and we predict how that process will operate across that range of inputs. And you can see here that the model predicted that the biocapacitance feed strategy was something that would be more robust and have a better performance than the fixed feed. And this was also this was confirmed experimentally. Okay, the last example for this part of the presentation is a single pass tangential flow filtration. And here a mechanistic model was calibrated, and then it was used to predict the behavior of this process during flow rate changes expected during manufacturing. So on the top right-hand side, we can see that the model was able to match the experimental values very well. And then the two contour plots on the bottom here show another very useful application of modeling during development, which is to explore the design space. So these were design space studies that were used to provide supporting evidence of process robustness. And we're going to come back later to this particular case study. So you may ask, well, how did you do that? How is this all done? So the models that we're using here are all based upon known science and engineering principles to predict the CQAs from the CPPs that influence them. And I think that the real difference to point out is that whereas a data-driven model or a statistical model may have tens of equations, these mechanistic and hybrid models, which we're using, typically have tens of thousands of equations. So they tend to come with a huge amount of knowledge that kind of built into the structure of the equations. But of course, they're correspondingly more complex to build. Now that's where Siemens comes in. So what we've done is we've implemented a established library of integrated mechanistic and hybrid mathematical models of all the different unit operations that you would usually expect to see in bioprocessing. So that's the equipment and the different phenomena. And this has been based upon the very best published science available. And we established this through our opinions, but also the opinions of leading academics and of our kind of partners and customers. So for all of these models, the equations are derived from the natural laws of physics, chemistry, and biology. So they contain really deep mathematical knowledge of the process, which significantly reduces the data requirements. If you think about it, what you're doing here is you're leveraging many people's research into how processes actually work. So with these models, the parameters of the models tend to have a physical meaning. So opposed to a data-driven model where the parameters of the models often you can't directly attribute to something going on in the process. So a model parameter for chromatography might be the capacity of the resin. Then the other thing which is quite different is that these equations are typically differential equations, or things are based upon differential equations. And so what we're doing here is we're predicting and simulating the process dynamics. And this is quite different to statistical models, which are often simply an input-output correlation. Now, where there are gaps in our scientific knowledge, which often occurs, right, with bioprocesses, this means that sometimes we can't derive mechanistic equations or first principle equations for the entire unit operation or bioprocess. And this is where we've seen that big advancement recently with the creation of this hybrid modeling approach, where we embed data-driven elements such as artificial neural networks to fill in the gaps in our knowledge. And we do this in a way that preserves as much as possible the benefits of the mechanistic model. So we really try to use mechanistic equations as much as possible, and then only bring in the data-driven part to cover the gaps in our knowledge. So these models are all available to use in sort of an out-of-the-box version in our GPROMS software. This is a software that's been developed and proven over many years that provides a really comprehensive process modeling and solution technology. So we configure models, and then we capture knowledge, or our customers capture knowledge, by importing their experimental data and calibrating the mechanistic or the hybrid model parameters. The other thing is that users can also modify the models as they wish. So custom modeling is something which is very popular in the software, building on top of the libraries that we've introduced ourselves. So people can introduce new equations, customized interfaces, and most often this is for particular, absorption isotherms or particular cell kinetic models that people have seen in papers and want to try out. Then once we have a validated model, of course, we utilize different solution techniques to try to exploit the model's predictive capability to create value. We do that through different things like simulation, global system analysis, and optimization, which is all part of the solution. Okay.
So before we continue, we'll go on to poll number 2 for everybody. So I'm going to go ahead and share that now. If you guys can go ahead and participate for us, give you a minute or so. All right, fantastic. Looks like we got our responses.
Okay, let's keep going. So, yeah, so far what we've considered is unit operation modeling in isolation. And this, of course, is very similar to how we develop processes using experimental approaches. Now, one of the things that's often overlooked when it comes to bioprocess digital twins is that process models can generate huge value by performing tasks which are not possible using an experimental approach, or just very difficult. And this could be due to the material requirements being prohibitive, or just the process complexity, the difficulty of building an end-to-end process in the lab. So end-to-end integration is a particular aspect of process modeling, which can generate a lot of value. So whilst most applications of modeling over the last decade have been limited to single unit operations, it's now possible to build integrated end-to-end bioprocess models. And the real value here is that the digital twin can be used offline to simulate, predict, and optimize the integrated and scaled up process before investing in the physical equipment at scale. And this enables organizations to build virtual plants and to test control strategies, look at disturbance rejection, look at operating scenarios in silico. So let's have a look at a case study here. So this is a case study where we have a continuous monoclonal antibody production process. And on the left-hand side of kind of the main picture, you can see the schematic, and on the right-hand side, you can see the process model, the full process model in GPROMS. So yeah, an end-to-end bioprocess model is configured in GPROMS to design and test the holistic control strategy of this process. And so the model is able to simulate the simultaneous and integrated operation of the full bioprocess. And so this includes a perfusion bioreactor to start with, followed by a protein A capture step. In this case, it's operating in a periodic countercurrent chromatography mode of 4 columns. This then goes through viral inactivation, a depth filter, before going to a cation exchange column and an anion exchange column. These are both, again, operated periodic countercurrent chromatography. We have a 3-column step and a 2-column step here. And then we have a number of nanofilter and a single pass TFF. So we were able to confirm that the model is able to predict this process. And then what it was used for is to predict and explore the behavior of the process after a transient disturbance event, both in terms of the magnitude of that event and the duration. And the timescale here gives insight on the residence time distribution of this process. So in this study, we can see how there was an unexpected aggregate production simulated, and the idea here is that this could be due to a deviation in the viral inactivation step, and we were able to use the model to explore how that deviation then affects the downstream process, and how does it move through that process. So you can see that this disturbance was introduced on the second day in this case study, and the model is predicting how the aggregate is then moving through the anion exchange column, and ultimately how it changes the concentration of the dimer and the monomer in the product concentration tank. So the outputs from the model here inform, or were intended to inform, on the material diversion strategy, allowing the customer to be able to divert product impacted by disturbance without impacting the product quality recovered in the batch. And ultimately, the vision here is to link this to real-time detection of critical process parameter deviations in the real process. So I thought I'd give you another quick example. So this one's a Siemens internal case study. It's a similar type of process, but here we can also see another example where, in this case, we have a perfusion bioreactor, and it's a bit more of an extreme case study. We're actually breaking a critical pump. So this is the pump which takes material out of the bioreactor, and we say it breaks for a day. So when this happens, we can't feed the perfusion bioreactor any longer, and we can no longer take spent media out of this bioreactor. So what happens is we have an accumulation of things like ammonia, which is toxic to cells, and this means we also have a depletion of nutrients, and it causes cells, or viable cells, to start to die, and then these dead cells start to lyse. So once we turn the pump back on again, we find that yes, the cells will return to a steady state, but what we find is that that cell death and lysis rates over that kind of 2-day period has caused an increase in aggregate and the release of host cell protein and DNA. And similar to that last case study, we can then track that as it goes down the process. So this is the protein A column, and we can see it takes a few days in order for that material to then kind of come back to a steady state. Now in this case, what we're doing is we're showing that the disturbance, whilst it's brought the capture step to a new steady state, there's actually no impact because the anion exchange column is robust enough to deal with that increase in aggregate. Okay, so let's talk about taking models online. So far, what we've considered have been case studies that have looked at offline applications, and what we are finding in general is that Siemens customers are increasingly looking to deploy models online for real-time applications. So when it comes to taking models online, all online deployment is underpinned by a validated predictive model of the unit operation or the process. And when we say online, end-to-end, so this presentation is about end-to-end bioprocess digital twins, we mean both the ability to model all of the bioprocess steps, but equally important is the ability to leverage investment in developing a model in one part of the life cycle many times over by reusing the same model that you've developed for R&D or engineering, but for digital operations. So when we deploy models, we build up the complexity in a sequential manner. And the first thing is that it's critical, obviously, that the model is accurately representing the process at a particular point in time. And so obviously we're bringing in a calibrated model of your process to start with. But what we can find is that if we only use this model and we don't update that model, it can drift away from what the process is doing in practice. So what we do is that rather than using the inputs kind of specified by the user, instead we now link that in real time to how the process is operating. And so if there's a flow rate change in the real process, that data is then sent directly to the model and the model is updated. Okay, so the other thing which we do is we perform something called state estimation. And this is where we use real-time data to continually update not only the inputs to the model, but we can also update the parameters that we need to calibrate in R&D, for example. So for this, GPROMS includes something called an extended Kalman filter or a moving horizon estimation. So what we do here is we can iteratively perform parameter estimation online using the real-time data. And this makes sure that when we are using a model online, it is accurately representing this process. So this is the first step. So in addition to training or tweaking the model when we're doing this state estimation, the other thing which it enables us to do is do something called soft sensing. So this is where we can leverage the real-time data combined with the detailed mathematical description of the phenomena to use the model to determine things that cannot be measured easily in real time. So what we can do is we can use the model to give us an indication of the cell health or resin aging or membrane fouling, something like this. So the next stage in building up this basically a digital twin is forecasting. And this is where we can use the model to predict what will happen in the future. This is particularly useful, of course, for bioprocesses, which tend to be quite long processes and are dynamic. So similar to that case study that we saw earlier, what we can do is we can use the model to predict, or the digital twin, to predict what will happen after a disturbance. We can predict the batch outcome and an alarm if we find that things aren't going to go the way we expect, which will allow operators to make interventions much earlier than waiting for something to happen. Then even something as basic as predicting how long it will take to complete steps can be quite valuable in bioprocessing, with obviously the variability often seen upstream changing tighter. We can actually use the digital twin to be able to understand at current flow rates, how long is it going to take us to be able to process that material in the downstream. And then lastly, we have advisory and we have autonomous. So in advisory mode, the digital twin can perform what-if analysis. So operators or people that are running this process are able to take the current state and to be able to punch in different ways of operating the process from that point onwards. And then the model and the digital twin is able to actually predict what is likely to happen. And of course, this can be very useful for people to understand what potentially can happen if they were to change something. We can also ask the digital twin to actually find the optimal operating conditions. So we can ask the model or the digital twin, how would you operate, or how should I be operating this in order to do things like maximize productivity or make sure that my viable cell concentration remains as high as possible for as long as possible. Now, this is in an advisory mode, so those recommendations are sent back to the operators, who can then make a decision on what they will do. The last is autonomous, which of course is when we close the loop and we take out the operators and we ask the digital twin to control the process, doing a similar thing as what we see in the advisory mode, apart from now when the digital twin comes up with the set points, those set points are sent directly back to the DCS system to implement. Okay, so for all of these different types of digital twin, we're using the same model. And so this allows us to leverage the investment made in one part of the life cycle many times over. So let's have a look at a case study. So here we see the same case study as earlier in the presentation. If you remember, this was a case study where the customer developed a model of a single pass tangential flow filtration step and then used it to explore the design space. So that same model was also utilized as an online solution or a bioprocess digital twin. So one of the big challenges of membrane filtration, or TFF, as you probably all know, is that fouling often degrades the process performance over time. And in this case, the step is concentrating a monoclonal antibody, but as the membrane fouls, there's a risk that the target concentration isn't going to be met. So in this work, the model is deployed online. It receives real-time pressure and flow rates. And then what the digital twin does is it acquires the data, it validates the data, and then it does a state estimation. And with the membrane, here we're estimating or soft sensing the membrane resistance in real time every 15 seconds. So this provides an accurate membrane health indicator for the operators, so they can understand what's actually going on inside that membrane. We can see from the graph on the left-hand side here that the model is able to accurately track the different pressures. And then on the right-hand side here, we see a maintenance forecast, which is performed with the model predicting how the membrane fouling process is going to proceed. And of course, this can help massively with decision making. So for those of you who are interested in this case study and would like to learn a bit more, in 1 week's time, I think it's next Wednesday as well, at a similar time, Merck are going to be presenting in a webinar. So we'll be providing some of the details. If you're interested in learning more, feel free to join up to that webinar. So how did we do this? So all GPROM's bioprocess digital twins are built upon the GPROM's digital applications platform. So this is a really robust integration platform that was developed back in 2016 and is now being used around the world with many successful case studies. And it's used for implementing models within operation systems. So the key features here include data exchange with external sources, so connecting to a DCS, a historian, or sending results to dashboards. It contains data validation modules, and corrective action can be taken if data is invalid. So we can define limits, we can define what to do if there's missing data. The platform is able to trigger model-based calculations, and it's able to schedule multiple calculations either to occur in sequence or in parallel. So with that Merck case study, which I just explained, what we have there is a state estimation calculation followed by a simulation. Now, if we take things and we do things which are closed loop, we might have state estimation followed by an optimization, where the optimization is performing a non-linear model predictive control type calculation. Of course, the other thing which is really key when it comes to taking these models online as bioprocess digital twins is system monitoring and recovery in the event of failure. So this platform has been built to be very robust and has a number of procedures in order to deal with situations which are unexpected. So it really provides a unified framework for sustainable and scalable deployment of models across organizations in manufacturing. Okay, so I'd like to wrap up now with a few final considerations and remarks on what we might expect in the future. So at Siemens, and I hope you've kind of seen this throughout the presentation, we really see bioprocess digital twins as a central evolving repository of knowledge in biopharmaceutical organizations. And we think it's really important to take an end-to-end and a unified approach to this type of technology. This enables organizations to implement sustainable modeling infrastructures so that they can leverage investments made in one part of the life cycle across the entire digital thread. So similar to how we have kind of platform manufacturing processes, we expect organizations to be curating platform digital process twins, recognizing, of course, that these solutions are going to need to continuously evolve by integrating the latest predictive science, experimental data, and know-how about your process.
Okay, and then I'm going to share the last poll, Ed, before we go to the next slide. Okay, if you can all just take a minute to go ahead and participate. All right, fantastic. Thank you so much.
Okay, so last few slides. So when it comes to the future, what do we expect to see? So we've spoken a bit about how industry has moved from predominantly offline modeling to now also taking models online, digital process twins. So one thing that you may have noticed is that the example that I showed you was a state estimation and a forecasting. This is what we're seeing a lot of examples of now. Now, at the moment, there have been no publicly presented examples, at least in biopharma or bioprocessing, for non-linear model predictive control in a closed loop format. So many organizations are working on this. So one of the things that we expect to see in the near future is going to be examples of this in industry. Now, this case study I'm showing here is from work presented by Janssen and Siemens and IFPAC. This was presented about a year and a half ago. And this is a real example of a non-linear model predictive controller, in this case for solvent swap optimization in synthetic pharma. So this was developed in GPROMS and implemented in a GMP facility at Janssen. And it resulted in a really significant reduction in costs, time, and also operator workload. So our customers in the biopharma industry are not very far behind. So certainly we expect to see more of these examples in the future. And then I think the last point is we've spoken about how industry has moved from predominantly unit operation modeling and R&D and engineering, but now using or also using end-to-end process models. And the value here is that we can perform tasks using models that are very difficult to do experimentally. What I kind of leave you with is a comment that I don't think we're very far away now from organizations making the same jump in manufacturing. So we expect to see in the future companies using end-to-end bioprocess digital twins to do things like soft sensing, monitoring, and real-time optimization of the entire bioprocess. So whereas previously we've been limited by science and sort of technologies that are available, I think recent advances now mean that this is entirely feasible. So this is much more of a matter of time. So an exciting time to be interested in this technology. Let's see where this goes. So with that, I'd like to thank everyone for joining today. And of course, I think we still have a bit of time now to answer any questions from the chat. So Angela, I think you might have one last poll and then we'll jump into the questions. I'll have a look, see what's come in.
Yes, and I did also want to share just one more thing with the audience as well. So before we go into that last poll, let me go ahead and share this with you. So we actually do have some hands-on trainings which are available on our website. So these are complimentary and they're self-paced. Just a little bit about these trainings. So once you do register, you'll have 24/7 access to these trainings. You can log in whenever. And we do have quite the comprehensive catalog as well, which will just help you get to know the software a little bit better and be able to just learn it at your own pace. And then the last thing, so you'll see here, this is just the training website, which we will share after the webinar as well. So for anybody that's interested, you can go ahead and register and get access to the trainings. Okay, and then finally, as Ed mentioned before, we do have a webinar coming up next Wednesday on October 9th with speakers Dr. Faye Metzi-Guckel from Merck and Rob Taylor from Merck, as well as Ed. So we'd love to see you guys at that webinar as well. Okay, and then now I'm going to go ahead and just share this last poll before we get started on the Q&A. Okay. Give you guys a few seconds to go ahead and respond to that one. All right, fantastic. So it looks like we did get a few responses. So Ed, if we want to go ahead and get started on the Q&A, it looks like we have a good amount of questions here.
Yeah, you might have to stop me, Angela.
Yeah,
we have loads of questions. You keep track of time.
Sure.
Yeah, so yeah, thanks a lot everyone for the questions. There's some excellent questions, some very difficult questions, but I'll do my best. So okay, let me look through this. So one of the questions maybe to start with, so we've got one which is sort of, what are the main differences between these tools and other vendors? Can you kind of pull out what are the main differences? So hopefully some of this has come across during this presentation. So with our tools, the modeling tool which we have covers all of the bioprocess unit operations, and there are mechanistic and hybrid modeling approaches available. And I think we're one of the few that offers that full coverage of upstream, downstream, and drug product even. So we can model the entire bioprocess, or we can model sections of the bioprocess, and batch, semi-batch, and continuous processes are all included. This is one big difference. The other thing which I kind of said at one point is that the software is actually very customizable. So there's an equation-based approach, which means that you can customize the models without actually requiring any programming experience. And this is really great if you want to be implementing a few kind of tweaks to the model based upon something you've read in a paper or what have you. The other thing to comment on, I suppose, is the underlying technology, the algorithms which we have here. So gPROMS has now been around, I think, for sort of like 25 years, and there's a big team of people, developers, that are working on the software continuously. So the underlying algorithms tend to enable faster calculation. So we have global algorithms for things like validation, optimization, and GSA, global analysis. So we can parallelize and we can utilize high performance computing. And also the software includes a number of capabilities that are not typically available in other tools. So there's variance-based global sensitivity analysis, there's the design space explorer, which we showed a bit of, there's surrogate modeling, the state estimation, which we talked about, and then of course the nonlinear model predictive control capabilities. So I think that's the other big difference. So the gPROMS digital applications platform provides this very proven, robust solution for online deployment, which is already being used for many customer case studies. Okay, so I have another question. So this is a bit more related to the online. So could you elaborate on the interoperability between gPROMS and DCS systems like Delta V and Pi Server for data integration. So this is a great question because so with these tools, integration is possible with any existing automation system that you might have. So it doesn't have to be Siemens. So it can communicate via things like OPC UA or any other kind of Ethernet-based communication protocol that's common in OT environments. So the GDAP tool, which we discussed, has an external data manager. It manages communication with the external data servers, such as control systems or data historians, SQL databases, etc., etc. So this can be used to integrate with offline, near real-time, and equipment data as required. Okay, let's pick another one. So can you integrate with other tools, such as Python, MATLAB? So yeah, we hear this very often, so yes. So what we have is we have interfaces to both Python and MATLAB. We also have a general interface that allows you to connect with other tools, other software. So there's a dedicated interface to Python called Adapter-FO. This lets GPROMs call Python scripts, so you can actually run Python from inside GPROMs. And this is very useful often if you have a Python model for sort of part of the bioprocess. So a good example here is using a Python script to calculate a parameter for a unit operation. And you wanted to use this because you developed a statistical model for that outside of GPROMs. So something like KLA is a good example where you might have a Python model that's able to calculate KLA from a number of different equipment geometries or RPM or what have you. And then there's another tool, it's called GoPython, and so you can actually call GPROMs from Python itself, and then GPROMs will then send specified outputs, so basically results from the simulations back to Python. So yeah. Okay. There's one here on the end-to-end. So how does GPROMs deal with scheduling in an integrated model? So there are lots of approaches actually for scheduling. Now, the recommended and the simplest approach to model the scheduling of sort of batch bioprocesses or even continuous, I suppose. So when you start up or when you shut down continuous processes, is using the bioprocessing model library. So within the model libraries, there are things like tanks and control system models. And within those models, indeed actually within the bioreactor and the chromatography and the membranes and what have you, there's inbuilt scheduling options for each of the unit operation model. And really what's underpinning it, there are equations in each of the unit operation models that sort of automatically activate and deactivate as a function of time, or there's some type of trigger variable defined. So for example, you could have a trigger variable which is like the volume in a tank, so the tank starts to discharge once the volume exceeds a certain value. The other thing I suppose is that the model libraries also include these control system models, which can be coupled with sensors and sources. So if you have a control system for something like DO or for pH, then you can implement that control system, and the sensors and the controllers will be able to simulate that control system as if it was the real control system. And then there's also a much more advanced scheduling tool within GPROMs where you can define very detailed operating procedures. So sort of the recipes that define periods of operation. Okay, great questions. So I've got one bit simpler. How long does it take models to solve? So a single simulation usually ranges from seconds to minutes, something like that. So when you do large parameter estimations, what we're doing here is we're iteratively solving many simulations in order to kind of decide or work out what parameter value minimizes the difference between the experimental data and the model predictions. And so these calculations are more complex. They might take something even up to several hours for something like a bioreactor, which has lots of experiments, lots of data. It's similar with global system analysis. If you're performing hundreds or even thousands of individual simulations. Now, one thing is that GPROMs comes with this built-in parallelization. So with the calculations, you can decrease the time it takes to do those calculations proportionally to the number of cores that you have in your machine, and you can send this to high-performance computing clusters or even to something like Amazon Web Services to really speed up those calculations if desired. When you take things online, I think because bioprocesses often take lots of time, like hours to days to perform, the calculations can usually be performed well within the desired time frame. So that's not really ever a worry. Now, what we can do though is if models become too complex to solve quickly and reliably online, we have a tool in gPROMS to generate surrogate models. So we can generate things like PLS or ANN models based upon a detailed mechanistic model, and the ANN is able to solve much quicker and more robustly. Now, we don't use it for bioprocessing very often. We don't need to, but it can be useful. We often use it when we're kind of simultaneously modeling multiple manufacturing facilities in the energy and chemical sectors, for example. Okay, what's the time? Okay, we've still got a few minutes, which is great. There's a few more questions here. So, is it possible to build purely statistical models within gPROMS software? Yeah, so it's possible. So you can do statistical modeling in gPROMS, but to be honest with you, there's usually better external tools, Python or other tools, and they're normally better because they're entirely focused on statistical modeling. So because gPROMS is more of mechanistic and hybrid process models rather than statistical models, actually probably the best way, and what we make sure to offer, is there are definitely good ways to connect statistical models to gPROMS models, rather than try and reinvent the wheel, so to speak. Okay, there's another question on models here. Does gPROMS contain CFD models? No, so it's a good question. So gPROMS doesn't contain CFD. So gPROMS is really process models. Siemens has other software for CFD, StarCCM Plus, for example. Now, one thing though is that the real advantage of CFD is that CFD is very good at predicting fluid dynamics as a function of the geometry of a bioreactor, or something like, which is very irregular, like the shape of a stirring impeller, for example. And what you can do is actually you can connect gPROMS to the CFD model, and you can do this two ways. You can either do it by generating something called a reduced order model, or a surrogate model, from the CFD, and then you can link that to the gPROMS variable. KLA is a good example here. Or we can also do something called multi-zonal modeling in gPROMS, where we break up the unit operation into multiple zones in gPROMS. And so each of the zones then has its own mixing characteristics and the concentrations of the various species. And then what we do is we can use the CFD to determine the flow of material between the zones in the gPROMS model. So obviously CFD is quite computationally heavy. So typically what we do is we run CFD in advance, and then we kind of import that into gPROMS. But it is also possible to do a two-way connection where gPROMS is then able to send information on the zone back to the CFD. So it could change the viscosity or something, which would then update the CFD. But of course, that's more definitely for R&D, where there's more time available. You don't need to solve these models so quickly because the CFD will really slow down the calculation. Okay. Okay, very general question, which is very hard to answer, but how much data is required to calibrate these models? So with the software, it comes with guidance documentation for each unit operation model on the kind of general data requirements for model calibration, also sort of how to use the data. So in general, the data required to calibrate is always less than kind of an equivalent data-driven or statistical model. Now there's lots of factors that comes into play, right? Because it could be prior knowledge that reduces the data required, the model complexity. If you have a more complex model, you want it to predict more factors and responses, you need more data. The context of use is important. What kind of questions are we expecting the model to be able to answer? So the exact experimental data required depends on lots of factors. We need dynamic data most often. This is a big difference between statistical modeling and process modeling using these dynamic mechanistic models. So you've seen in the case studies that we're taking samples at regular intervals. Typically, our customers utilize process development data from scale-down lab experiments, and then they're able to transfer parameters to models of larger scale, because many of the parameters in these mechanistic models are scale independent. So you only need to really re-estimate or tune the scale-dependent parameters, which also reduces data requirements. And yeah, maybe just generally, for chromatography, you've seen it could be as low as 3 experiments. For bioreactors, you've seen today how sort of you can get a fairly reasonable prediction using something like 10 experiments covering quite a wide range of conditions. We have another question which is about sort of how many inputs and outputs are we considering. So with a bioreactor model, you typically are going to be considering maybe up to sort of like 15 different species when it comes to these models. You might also consider temperature shifts. You might consider the initial seeding density. So you can tend to have quite a large number of factors, and it means the models you might have something like 20, 30 different parameters that we need to estimate. Okay, and then for something like a TFF or a UFDF, you could have something like 10 experiments to calibrate your model. Okay, all right, so a couple last questions. I know we've got like only a few minutes. So do your models consider molecular structure? So in general, no. We've implemented models that don't consider the molecule structure. So you do need to calibrate the models per molecule. We are finding though that models can sometimes be similar between molecules, especially if you've got the same cell line, for example. So this opens that possibility to build these platform models where you have a base structure and then you tweak those models for each new asset. For some of the absorption isotherms in chromatography, you've got charge, which is related to molecule structure. For TFF, there's a model you have which predicts the effect of Donon partitioning on the final buffer composition. And of course, like KLA, not KLA, sorry, pKa of the molecule is related to amino acid sequence, so that's useful. And then, okay, one last question before we finish up, which is probably the hardest one. Left the worst to last. Can you comment on regulatory considerations when using models in filings? It actually is not so hard. We have good experience on this. So what we recommend is something which is closely aligned with the regulatory guidance on this, and it's also something that we've presented on with the FDA actually a few years ago, and it kind of aligns with the principles of model qualification, verification, and validation. So this has been set out in sort of books and literature on scientific computing. And essentially what we do is when you want to use something and when you want to file a model, we recommend taking a risk-informed credibility assessment framework to kind of determine how accurate does the model need to be, what's the validation space, before we perform the experiments to calibrate and validate it. And what you need to start with is what's the question the model needs to answer, or what's the concern that's being addressed. This then establishes the role and the scope of the model in addressing that question. And so what you need to do is provide an explanation of how the model or the digital twin will be used to answer the question, and also what are the other sources of evidence that we use as part of that decision. And that really helps, because if you have lots of sources that are informing a decision, it will reduce the importance of the model and the risk of the model. Of course, yeah, risk is a big part of it. So we need to know what's the consequence of making a wrong decision and what's the severity of an adverse outcome if you have an incorrect decision. So all of this kind of leads into these credibility risk assessment. And there's sort of these 15 categories where you have to justify the equations, you have to provide evidence that they've been implemented correctly in code and that they're being solved accurately. And then last but not least, we need to provide evidence that the model is being calibrated properly. So one of the, obviously the key advantages of using a commercial software like we offer at Siemens is it provides you with assurance that the first two categories are met, and then the model calibration approach is suitable. Okay, so yeah, great question. I think I'll leave it at that and pass it back over to Angela.
Yeah, thanks, Ed.
Thanks very much.
I did just want to address one question that I did see come in a few times about the recording of the webinar. So within the hour, you will receive an email with a link to the recording so you can view the webinar again on your own time. And with that, I just want to say thanks to our presenter, Ed, again, and thanks for everyone for joining today. We will look forward to hearing from you soon. Thanks, everyone.
Thank you for watching. To learn more about this topic, check out the links in the description. And if you enjoyed what you saw, like the video and subscribe to the channel.